So you have asked us to illustrate and describe meiosis. So I went ahead and drew out the outlines just because it's such a lengthy process to describe and even write out. So there are two phases to meiosis, meiosis 1 and meiosis 2. So I'll start with meiosis 1. The very first phase is interphase 1. And in interphase 1, the chromatin is still stringy. It is not condensed yet. You can hardly see it or distinguish it. And the centrosome has been copied, so there are two centrosomes, each with pairs of centrioles. And this is where microtubules extend from. And actually, the mitotic spindle sort of starts to appear in this phase. Next is prophase 1. Now within meiosis, prophase is a very lengthy process, so I'm going to describe it and then I'm going to sort of draw it out for you. So prophase actually has 5 different stages to it. Those stages are leptotene, zygotene, pachytene, diplotene, and diakinesis. But I'm just going to separate them into early, mid, and late prophase to save time. Leptotene is a stage where the condensation of chromatin fibers form into long thread-like structures similar to mitosis. Zygotene then condenses those fibers to make individual chromosomes, and they're actually distinguishable in that stage. Homologous chromosomes become closely paired with each other via the process of synapsis, and this forms bivalence. This is considered genetically significant because the close proximity between the homologous chromosomes allows for crossing over, which is DNA exchange, and this accounts for genetic recombination. So this crossing over is a very big part of genetic variation within meiosis. 2 chromosomes of each homologous pair remain attached by connections known as chiasmata. These are situated in regions where homologous pairs have exchanged DNA segments and have hence provided visual evidence that crossing over has occurred between the 2 chromatids. The centromeres of the homologous structures begin to separate, eventually leaving the chiasmata the only remaining attachments between the homologous pairs. The nuclear envelope begins to break down and marks the end of prophase. So I'm going to draw this out for you. So in early prophase, the chromatin starts to condense, and the centromeres, where is my blue? Start to separate and expel the mitotic spindle. And then in mid-prophase, they condense into distinguishable structures and pair together into bivalence. And this is where some crossing over occurs. Again, allowing for genetic variation. And the centromeres start to separate even more. More mitotic spindle. And then in late prophase, in late prophase, the pairs separate, leaving chiasmata where the DNA crossing over happened. The nuclear envelope starts to disappear, again marking the end of prophase. And the centromeres are on opposite poles, close to getting to opposite poles, and have released mitotic spindles. So, now that prophase is ended, next is metaphase 1, where bivalents such as kinetochores, or bivalents attach to kinetochores to spindle microtubules and migrate to the spindle equator. So I'll draw that out for you. I'll draw a little bit more. Also, with their chiasmatas, where DNA crossing over happened, the centromeres and the meiotic spindle attached to the kinetochores. So, where they're lined up is called the spindle equator, and centrioles at the opposite poles of the cells with meiotic spindles extend to attach to the kinetochores. So then we have anaphase 1, where I'll explain it. In anaphase 1, homologous chromosomes separate and move to opposite poles, while the sister chromatids remain together. So that's another really important thing about meiosis 1, is that they separate with the chromatids still together. They do not separate to opposite poles yet in meiosis 1. And each pole receives a haploid set of chromosomes. The chromosomes line up in independent assortment, which is another factor of genetic variation. So the chromosomes line up in random, lead into genetic variation, like I said. There's no specific way they line up. So I'll draw this out. The whole pair, also with their chiasmatas, can't forget that. Centromeres. And the whole pair is being separated to the poles. And then we have telophase 1, where chromosomes complete their move to opposite poles of the cell. Each pole of the cell has a full set of chromosomes together. A membrane forms or starts to form around each set of chromosomes to create 2 new nuclei. The single cell pinches in the middle to form separate daughter cells, which is cytokinesis, and each contains a full set of chromosomes. So I'll write this out for you. Again, independent assortment decides this. There's no specific way that these lined up to be separated. Centromere. And then cytokinesis starts to separate the two into 2 separate cells, and a nuclear envelope starts to form again. So we move on to meiosis 2. I actually started to draw it out. I thought I was recording, but I wasn't. And the very first stage of meiosis 2 is prophase 2, where there are 2 daughter cells, each with 23 chromosomes that have started to condense. The membrane around the nucleus starts to dissolve away, releasing the chromosomes. So you can see that it started to dissolve. And then the centrioles duplicate, so there's 2, and a meiotic spindle starts to form, as you can see. Next is metaphase 2, where the sister chromatids line up in the equator, the spindle equator, and the meiotic spindle from the poles attached to the kinetochores. Again, also, can't forget about the chiasmatas, where the DNA has crossed over, creating genetic variability. And then is anaphase 2. So in anaphase 2, sister chromatids are pulled to opposite poles due to the action of meiotic spindle, again from motor proteins, and they're separated chromatids are now individual chromosomes, like we saw in mitosis. So let me draw this out. Chiasmatas. Genetic variation is a big part of meiosis, so we can't forget about this. Being separated. With meiotic spindle, driven by motor proteins. So there's that. And then lastly is telophase 2, where chromosomes are completely moved to opposite poles. Each pole has a full set of chromosomes. A membrane starts to reform around each set of chromosomes, creating two nuclei. Cytokinesis starts to separate into 4 granddaughter cells, each with half a set of chromosomes. So, let me draw this out. They start to decondense after this into long fibers again. So it creates 4 granddaughter cells.